You'll notice that there's two people who haven't yet spoken to you all, so I just want to have them introduce themselves, but we're also joined by Nir Hacohen from the Bird and MGH and Jean Herelle from Crunch Foundation. And you can just give introductions to yourselves since you're.
Sure. I'm a faculty member here and at Mass General and study cancer immunology and systems immunology, use a lot of computational approaches to look at these problems and try to get a systems level view.
Hello everyone, I'm Jean Herelle. I'm the founder of CrunchDAO, Crunch Lab, so the platform that has been organizing this challenge with Caroline Uhler and all the teams here. So really proud to be here and really proud of the result that I've been displaying.
Oh, I don't think you did. Yeah, it was just the two different. Okay, so yeah, so we've kind of prepared a set of questions here for the panel, and some are, maybe more suited for one person or another, but everyone is feel free to jump in and give your feedback or follow-up questions. So we'll start with what we talked about, which is one of the hardest things, which was defining these problems that we're working on. So, research is usually this open-ended discovery process where you're either testing a hypothesis or generating new hypotheses. So how do you take a complex biological question and distill it into these sort of three tasks that we were seeing in each of the crunches? And I guess I'll start with Nir and Caroline's views on this.
Sure. So I think each problem has a history and a trajectory. When we do the actual challenge, there's a moment in time. And the question I think that we should always address in that moment in time is, what is the most important unanswered question at that moment? And be able to just conceptualize that. And so a lot of what we do, and we have an open problem, is of course we have to screen hypotheses. That is what we do anyway in the lab. So I think the way I see it is that the analogy is, we've got this open problem, we're stuck, we don't know the answer. And so if we can conceptualize that problem, put that into words and say, here are the different problems that we need to solve today, may be different than 3 years from now. And I think we need to basically bring two things together. One is, what's the biological problem that needs to be solved? And then what's the paradigm problem that we're trying to solve more generally? And if we can bring those two together, I think then we have a really good challenge, because it's both biologically motivated for that moment where you have an unanswered question. And if you solve it, it goes way beyond the biology that we just introduced. And I think that's what we tried to do in the first challenge, is to get those two points correct for that moment in time. It's still an unsolved problem today, a few years later. So it's not that that the world has solved it. So we gave a good, hard problem for sure.
In addition to that, it's about then defining the metrics that would tell you whether you are actually going into the right direction for your biological problem, where I think we spent a lot of time on. And that's always a big discussion that has to be done in collaboration, so that we really come up with meaningful metrics.
And if I can add one thing on this, I think I've been extremely impressed by how hard this problem have been and how ambitious the team here have been with these problems. And we had different problems on the platform. And it's very important to bring these communities to the bleeding edge of that technologies, and where you guys are actually stuck. There are some tournaments or some hackathons are sometimes about, what could be fun to work out on, or what could be bringing as many people as possible. But here, these three challenges have been extremely challenging, I think, and extremely interesting. That's why it's been so successful. So I hope some people will find solutions very soon. But I think that defining this problem is very, very important. And thank you for coming with such a challenging problem to the Crunch community. It's been great.
Great. Yeah. And I think, in defining the problem, Caroline alluded to that part of this is like defining the metrics, right? So in, machine learning, what we see a lot is that, there's very quantifiable metrics. We want low RMSE or high AUC, but biology tends to resist these obvious metrics, and people want to cure a disease, make a better immunotherapy, and how do you quantify performance there? So there's this kind of not tension, but different objectives between a very quantifiable metric where your machine learning model produces some numbers, and a biological metric where you've moved the biological system in a direction you care about. So we've all thought about this, and probably people here don't know, but I think we talked for 3 or 4 months on how to define T cell states, just to give you a sense of like how some of these things can be very challenging. So just curious about how people view defining these metrics. And maybe Jachi, you go for it.
I guess I can start.
Yeah.
I think the way I think about it is we work backward. We work backward in the sense that we first think about once we have these models, what do we want these models to do? And in our case, we're interested in, or like broadly speaking, making better therapies. And we decided that a way to quantify that is through proportion of T cell state, which you mentioned we spent a lot of months discussing how to define those states. We went back and forth looking at different data normalization and everything, and finding different marker genes. So it took a long time. And then because the eventual success is defined in that space, so we sort of grounded our metrics at that level of abstraction. By the level of abstraction, I meant that because we have single cell RNA-seq, we could measure, we could do different kinds of metrics. We can do RMSE in terms of bulk prediction, or we can do single cell level prediction like MMD, as we did in the third challenge, or we can do differential expression or finding DE genes, everything like that. But we decided to go to the cell state level because that's the level that's closest to what we want the models to do, which arguably is of the most useful biological utility. But you could argue that maybe this level of abstraction wouldn't allow us to transfer the model to some other task where we might not be interested in states or something like that, which is totally fair. But I don't think, so maybe we heard a lot of talks. I don't know if we collected enough data to be able to say that any models trained here can be transferred to another totally different contexts. So we might as well just focus on this problem, which is hard enough, and define the metrics there.
So that's great, a great segue,
Yeah.
Jachi. So like you bring up that, we were trying to, get T cell states and shift them here and there. And then somebody else is making perturbations in a different system, different tissue, different cells, and they want a different biological objective function. So when you have that, like, how do you actually develop a model that can do all these biological tasks? Do you think you actually can still make foundation models that can do everything, or do you need these task-specific models? Like, how do you think about that? And maybe like Daniel and Shijun also, because you guys worked on a very similar task, which is predicting held out perturbations, but a very different biological objective. So like, how do those kind of conflicts across the different cells and tissues affect model generation?
I think I just have a very simple plot in my mind where we collect data across the time, and there's like eventually going to maybe plateau or something, or like grow. And maybe at like the low data regime is maybe where we sit as of now for perturbation prediction. And there, a lot of domain knowledge, I think, is very important, and like a specific model that captures inductive biases, prior knowledge on the genes are very important. But I think as we collect more and more data, there might be foundation model that have this emergent property that can transfer across different contexts. But you were saying, I guess I'll just let others speak for a moment. Yeah.
Yeah. So I think you mentioned two questions. First is related to the scoring matrix. I have two thoughts. One is I think the scoring matrices are quite hard, especially in the field of spatial transcriptomics, where it's quite exploratory and open-ended. So different tasks has different like kind of goals. But for the spatial challenge, we need to have one fair kind of comparisons. So that in that case, kind of one potential risk is the method try to engineer or optimize on one scoring matrix may not be optimized for other tasks that seemingly relevant. But I think that's also the benefits for our spatial challenge, that we have experimental validations on the out of distribution samples so that we can think about like if a method perform very good on one scoring matrix, how it perform on something that seems similar but not exactly the same. So that's one thought. Another thing is quite interesting is during the machine learning challenge, actually we kind of include another scoring matrix in the middle of the computations. So the interesting thing happened is we have some community kind of forums, and we have some interactions and discussions. And some of the participants proposed that probably, say, a gene-wide Spearman correlation could be better suited for the second spatial challenge. So after our kind of internal evaluation and discussions, we think that, yeah, that's probably a good suggestions. So for our spatial challenge, we kind of try to also kind of incorporate another scoring matrix as a final benchmarking or leaderboard calculations.
Great. Thanks, Lujan. And Daniel, actually, I'd love your perspective because you're the, I guess, other than Nir, the biggest biologist on this panel. So how do you view these? Second. We're all second to Nir.
There you go. Yeah. I think what's coming through clearly here is that there's still just no substitute for people actually looking and caring and thinking about the biology in each of these systems. I think that's what's made these machine learning challenges successful, is that people are actually sitting with the data and figuring out, okay, what are the best metrics for this, right? I don't doubt that there's a point that we've generated, enough data and enough cell lines within enough genetic backgrounds across enough environments, to be able to sort of not have to do that de novo every single time. I just think we're like a really long way from that. So we don't have to worry about it too much yet, I think. Yeah.
Yeah, maybe I can add on top of that. So I think in the obesity challenge, we definitely have two sets of evaluation metrics. One is more about the prediction, like accuracy, more like the IMSE that we talk about. We also have the second set that is trying to capture more biological questions of interest, and it's being made based on a lot of discussions with Daniel. And what we have observed so far is that the methods that perform well in the first set of metrics are not necessarily the ones that perform very well in the second metrics. So my personal view of this is like the more ML metrics can try to get an assessment of how good the model predicts, but then the more bio metrics is actually like tell us whether those predictions are actually useful. And for now, I don't see like the trend very consistent in these two separate things. So I think my personal take is that we still need a lot of collaborations and discussing with the biologists to figure out what's the correct things to do.
Great. So lots of good perspectives there. So shifting gears a bit, just talking about, running a competition in kind of an academic setting. So the typical way we know a lot of us do science is we collect our data using some new technology, we build a model, we learn something, and we publish it. And this is really different because actually, like, all this data has not been published yet. Everybody had to agree to put it out there. And so that opens you up to people finding artifacts in the data, or they find that they build a model that's much better than anything you have. So how do, like Nir and Caroline, like, how do you think about some of those issues? Are they concerns, or are they benefits, bad things?
Well, I mean, I think you should design it so it's only benefiting. So, and I think we have, at least for our perspective, we have. So in our project, at least, we certainly focused on having a good collection of genes that weren't necessarily the ones that we're like studying in the lab this minute, like, so that we have a broader collection than our kind of favorite thing this minute. And that, I think, opened us up to new directions and potential discoveries that we could follow up later. So it's both creating the right data set that's not competing with the exact thing you're doing in the next paper, but similar enough. And that's what we did. And then also thinking, if this were successful, what kind of biology would it lead to the community or us to follow up? And I think if you do that carefully, I mean, we did have to talk about it at the beginning and try to figure out what is the right kind of data set that wouldn't compete with ourselves, because we are going to just release it to the whole world immediately before. But I don't think it's hard to do with high throughput experiments. There's enough space there for, in most cases, that it doesn't compete, fortunately.
And I guess on our side, I mean, we never think that we have the best model. We always want to invite, others to tell us where do we actually stand, and invite others with completely different ideas. In particular, if we're thinking about, machine learning experts who work on completely different problems, and invite them in. And I'm sure we can all learn something from them. And that will just, I think all of us as researchers, we want to move the field forward, and not just like one method forward. So we can only gain from having more and more people come into the field of like trying to work on really hard biomedical problems.
Actually, maybe I'll just add one thing. I think this has been really fun because this whole thing of like you generate a data set, it has to like at least be or appear perfect, and then you publish it, and then like there's this place for that obviously in academia, but this sort of competition where it's like we generate data, experimental data is never perfect, machine learning models are never perfect, right? And we put them out there and we sort of all work together towards solving, a common problem that we're all interested in, I think is sort of a nice way to do it. Kind of removes that, fictitious idea that like actually we're trying to generate some, perfect data, right? Like somebody finding something wrong with your data is actually a good thing. Then you can fix it on the next round, right? So.
Yeah, those are all really interesting points, and I agree. It's kind of different from like say where we had like the PDB, and you're solving protein structures, and they kind of stayed roughly the same. I know technologies might have gone better, but a lot of the genomics technology is kind of a moving target, and we're getting better and better at these measurements. So I think that kind of just openness to things getting better is important. A little different in terms of data. Okay, so then I also want to talk about the people who actually enter these challenges, and this is a question that I think Jean will have a lot of kind of ideas on here. But so we've gotten participants from 60 or more countries. Are there surprises about the backgrounds of the participants? Do you think we're drawing in new people to biology? Does the diversity of thought help lead to diverse solutions and problems?
Yeah, absolutely. So I think there is a lot of people that are really interested from the machine learning industry that would like to come and work on the problem that you guys are working on. And one of the main barriers is actually being able to work on some data, right? And so when you have been releasing these data sets and coming with also a problem statement, this is the first step to inviting people into your world, because the problem statement needs to come from expert. They are not expert. Most of them are not coming from that field. But without the right problem statement, they're going to run this kind of gradient descent, kind of human gradient descent that happened for like 6 months. People are improving on each other, talk to each other, build team, talk on the forums, have some ideas about what did you do, how do you end up on the tops, this information kind of leak, and then more people knows about it, and then slowly iterate. And so in this middle of this mix, we have people coming from all over all over engineering, computer science, statistics, life science, physicists, and they all come with their own methods, right? Physicists love the kernel methods. Mathematicians do just throw some neural nets at the problem, without even understanding it. I just need an objective function, which is also what you were just talking about, wrong metrics. As an ML engineer, the metrics is going to end up in the objective function day one. They're going to look at this metrics, try to make the model iterate and learn, and iterate on that metric. So all these guys come with different methods, and all these methods eventually fight one word that I've been hearing a lot for the past days, and we kind of create this big basket that we call overfitting, right? We call it overfitting. What is overfitting? It's actually failing to model things properly. There's many reasons to end up overfitting. And so when you have all these methods together, you get a chance at a regularization effect, which is just, okay, everybody is performing, but out of some are good, some are less good. And I think we've seen on the graph, so like 3 teams are on the top. The 3 teams used 3 different methods. And in these 3 different methods, there is an opportunity of regularizing your results and eventually limiting overfitting. That's one of the effects that I love in this competition, is that you can build kind of meta model, a kind of ensembles of models, which is widely used in AI, by the way. And in ML, you guys are building models, so you end up stacking or doing all these kind of methods to have different models together. So this can happen at a second level because everybody is building meta models or ensemble of models, and then you end up doing an ensemble of the ensemble of models. And it's actually deeper than that because everyone is using also LLMs on the thing. So they are using LLMs to find solutions that they then stack together that then you eventually end up stacking together. So we don't realize how much intelligence end up at the end of a 6 months work and a 6 months crunch like that. And I think this is really what you want to see. And so sometimes you happen in the data, sometimes you find something in the modeling, sometimes you find something in the metric. But I think there were a presentation earlier with the 3 pillars with data, validation, and models, right? So the crunch community or these kind of tournaments happen at the model, the last step. Like we need data and validation to be set up properly, and then model innovations happen behind.
Yeah, and I know like Jachi has done a lot of thinking about this like meta model, like taking the best features of all the participants' models and understanding what they learned and how they can be combined into something better. So Jachi, do you have any feelings on how that's going?
So combining those gene features, we've done it in a very simple way, and we see a little bit marginal kind of benefit, but not huge. I think there's like still open research going on in terms of how to best combine prediction coming from different perturbational models. You can imagine you can do it in the gene feature space, you can do it in the output space, so on and so forth. We are doing some ongoing explorations, so hopefully can share some results in a few weeks or months.
It's a very, and it's very difficult. I've been in that space as well after the competition when you try to ensemble things together. It's very hard because it's some very noisy problem. But I think what we've seen is not necessarily an improve in performance, but an improve of generalization. It's really this overfitting risk that you remove. So maybe you're a little bit less specific. You won't have the top accuracy in the model, but the model will be more the ensemble, the final ensembling of different methods end up being better at generalizing, which is important out of sample. And that's really, I think, what you can find it, but it's difficult.
Great. And I just want to close the loop on the human side of this with the participants. So like Jachi and Lujen, you interacted a lot with the participants on the forums. Were there any surprising behaviors, both like good and bad? Like I know on a good side, there were people who were very enthusiastic and asking lots of questions and wanting to understand how things work. But what did you observe? Or just some of the surprising things? Maybe we'll start with Jachi though. I know she's dealt with this a lot.
A little bit. Okay, I can share a backstory, which is like, I guess, maybe also early on from my background training, I probably was like not realizing how hard it is to perform an experiment and how much effort needs to be put in. There are like pilot screens, and Daniel can speak a lot to everything you need to prepare all the sequences, libraries, and it takes so long to do the experiments and do QC and eventually annotating the data to get out the result. So I think at some point we need to explain to the participant why we need to take a few months in order to get the experimental result. But it's good that some participants came in with wet lab experiences, and they actually explained for us. So eventually it's a happy ending. And that's what I can speak to, like a mix of bad and good story, I guess.
Yeah, it was funny to get emails asking, "Are you done your experiments?" And it was like, "You're not the PI, but you act like it." It was very entertaining. But yeah, but it was also the enthusiasm of like, "Hey, did I get this right? Was my prediction right?" Wanting to know, did it translate? So it was very interesting. Yeah. Lujen, also you dealt with a lot of people in the crunch. So any?
Yeah, I generally think it's quite beneficial to discuss with the participants, especially related to like how could we have a quite fair evaluations, because initially kind of we may start from say computational biology perspective, certain evaluation would be quite ideal. But then from interaction with the participants, where they have certain other kind of expertise in other field, they may think certain evaluation or scoring mechanisms may be kind of more kind of fair to the other type of consideration or something. So I think generally that's quite beneficial, that interacting with the people.
Jean, you want to say something?
Yeah, I think you realize that some people came and they didn't know what was a cell. And you try to do like this is such a complex thing. And they're like, oh yeah, but can you technically define what is a cell? And I think you've been an amazing job in doing these courses, and Caroline, because I think they were really needed because people were coming from far, and really from far. I mean, which is sometimes the people didn't study that, or they studied, biology long time ago at school, and they are computer scientists for like many years. They have to like restart from zero. And so some of the discussion were around that. Some others were around, people are always in the community when they care a lot about something. They're going to be after you every day in the forum by email, but it's really because they care. And they spend a lot of time, so they're excited about the result. And these results takes time to get validated, but they understand now. I think that they're getting used to it.
Great. So we have like about 15 minutes. So I wanted to give people in the audience time to ask this great panel some questions, anything related to competition. So if you'd like to ask a question, just raise your hand or yell at me and I'll come over with a mic. Yeah. Thanks.
Yeah, so we've heard already today and yesterday that often when it comes to perturbation prediction, simple methods still are quite competitive compared to complex foundation models. So I wanted to ask, what has your experience been in these challenges so far? How well do simple methods do? Are they usually amongst the best performing ones? And are you taking any steps when you come up with new challenges to make sure you focus on problems where we don't yet have simple solutions that do pretty well?
Just take one since I know you will like it. So there was a causal group who had never done anything biological who ended up in the top 10 in our first challenge. So it is very relevant to get in people with, completely new backgrounds. And so that was certainly not a simple method. That was, really one coming from a top causal group. So we definitely have surprises and, that's not always the simplest methods that do well, but many simple methods do well, and you guys can talk to that as well.
I think back when we were launching the challenge, which was maybe early 2023, there weren't any foundation model in biology. I think Gene Former came out May 2023. But there were like gene features derived from pre-trained models on large single-cell RNA-seq data, and those end up in the top 10. So they provide some useful information, I guess. But I think in the third challenge, maybe there are participants who are using foundation models, if it's possible to share.
Yeah, maybe I can share a bit more. I think the issue here might be that a lot of biological questions, they are highly context specific. But then at the same time, foundation models, they are very promising in terms of integrating different data sets from like very different sources. But then in order to make such foundation models to be highly helpful in terms of specific tasks, there might still be like some more careful design in terms of the algorithms, because it might not be always the case that you throw as much information as a big model and that's always turns out to be the most helpful. But I would say I still think foundation model is a very promising way, and we do see like some people, they are using the representations from the large-scale foundation models to boost their performance, and they're also one of the top performers in our current challenge.
And just adding for the spatial challenge, basically all of the top methods use large-scale pathology foundation models. So we only had one group who didn't, who ended up in the top 10.
Yeah, that's right. I think for that virtual spatial transcriptomics topics, I think I feel it's better to use the foundation models. Otherwise, you need to really train a large models from scratch. And that may kind of, that may not really kind of generalize to different like imaging conditions or so on and so forth. And for the third part, like proposed gene panels for the spatial autoimmune disease challenge, although that kind of topic is not directly related to the foundation models, but using LLM is quite beneficial for refining the gene list. Like traditionally, it may rely on us to kind of read the literatures and come up with some genes. But I think that's still very important to really understand the field. But at the same time, if we rely on, get some help from those ChatGPT or those kind of chatting LLM, that will kind of give us something that we haven't really thought about, and as some new kind of thing that we can evaluate in the experiments, and this may kind of have some benefits.
Maybe I can add one thing. I think there is also a probabilistic approach you can take to solve that problem. There is many simple ways to solve that problem, plus a complex way. And we've seen some presentation where you may end up in a situation where you have maybe 1,500,000 possible solutions that then you need to synthesize in the wet lab. And if you look at the problem from a probabilistic standpoint, then you have probability distribution from all the models. And then you can say, okay, everybody agrees, foundation model, simple solutions, and everybody agrees on that one in particular. So if we didn't test it, there's higher probability for that things to be interesting.
Anybody else have questions? Just raise your hand. I'll come over. I think we still have a few minutes, so we could ask one or two more questions of you. Or are you done? Okay, let's go for it. So I think so we talked about models, but also like the data. So this general question of like, how do you know you have enough data to really solve a problem? So I guess it, as an example for CASP, it turned out the PDB was in a sense enough data to solve that problem, or at least a defined part of the protein folding problem. So if we think about, virtual cell and cellular perturbations, how far do you think we are from having enough data to solve, specific problems like T cell states or, changing adipocyte states? Like, where do you think we are in that? Maybe Nir has.
Well, first on the protein design problem, we still don't have enough data. We're good at folding proteins that are in the world, but we're not actually very good at still folding proteins that are not novel, variable and so forth. So, and I don't think, having watched that field in the last couple of years, I don't think we actually know how much data we need there in order to correctly de novo design proteins yet, even though, that is the goal. So I don't have a good sense, because I think in the protein perturbation, the gene perturbation problem, I think no one has gotten to a high enough quality of success to try to predict that curve. That's my intuition at this point. But I think that, given that the K562 and the other ones where they're, from Jonathan Weissman's lab, where there's enough data, or there's a lot of data, still the problem of prediction is still hard. Maybe you guys who have worked on it can comment more about where you think that is. Maybe you have you were going to say that.
No, unfortunately, I cannot answer to this level. But the data is obviously, a big factor in modeling. Like in the 3 pillars again of your presentation earlier, it was the data come first. So of course, there is only as much we can do. But you guys should answer from the CAS standpoint.
Yeah, so I guess, going back to the CAS problem, you sort of don't have enough data until you do. And then, right, like that was a big surprise. No one was expecting AlphaFold to come out of nowhere, right? So I think that's it's sort of the right convergence between having the right data sets and the right tools at the right time. For the virtual cell, not even close, I think, for sure. The data sets that you're talking about are K562s, like these are cancer cell lines. We haven't even scratched the surface on what we need to be looking at here. But I think the machine learning challenge is kind of fun because we've sort of set this up with the sort of lag in the loop system where we're generating the data that we think that we need to answer the problem that we've set out to solve, right? And so at the end of like our machine learning challenge, I think we'll have tested 500 perturbations, tops, right? And this is like across all single genes and pairs. So this is really a tiny experiment, whether we find anything out of it. But let's say we do, right? Like we won't have tested this whole space. I think that's sort of an interesting way of thinking about this as well, where you have a predefined problem. You can sort of set out to generate the specific data you need to answer that. But yeah, as far as these large models go, I think like very far, probably. Yeah.
Yeah, that's a general sort of problem as to whether there's a benefit from just collecting a lot of data versus collecting data that's specific to the problem you really want.
The right data.
The right data, yeah. So we have time for one more question. So I don't know who raised their hand first. Okay.
I was curious whether in your research or in your modeling, do you take in consideration of the accuracy and error rate? Fortunately, and, in our medical AI, for instance, like digital pathology, or, medical imaging, the accuracy rate range from 80 to 95%, but we still have some error rate could up to 20%. So when you put a poster or when you present your model or the foundation model using the neural network and the data training, do you also state how accuracy or how precise when you run your model?
I mean, I can also answer that. It's just we always report all accuracy metrics and everything's very transparent, and we're never, I mean, medical AI, it's a lot more advanced in terms of labeling images. Like the accuracy in these problems is much lower. So there's still a long ways to go. Yeah. Maybe I can okay, we can do one quick question.
I guess my question is about drug response prediction. Machine learning models, a lot of these models are built for different cancer types and different diseases. So, but a lot of these results are heterogeneous. For example, for prostate cancer, late stage detection, if there is one drug that is given for like state of the art for metastatic castration resistant prostate cancer, the responses are heterogeneous. So one is like if you're building these models and you're looking at the drug resistance and sensitivity, how would you see this? How would you know the data with respect to the different genetic variations and biomarkers? And how do you also look at what kind of a clinical data to integrate and to make an assessment?
Maybe I'll take that to round up this panel, because I think that's this is now a completely different problem. So we would start off with starting to think again, what could be a new challenge that we actually set out? This could be a very interesting one. And then we start thinking about, do we have in-house data generation capabilities? Which disease would we look at? Where do we have experts within the Broad where we could actually come up with a meaningful challenge like this? And then try from a very broad question, like what you just asked, to actually boil it down to a very precise question, but that is still very relevant from, for a biomedical, for the bigger question that you're kind of asking, and then start working together towards, generating the data set and coming up with the right questions. So
great. So that's a great ending to the panel discussion. Thanks to all the panel members, and hopefully everybody here got a sense of, how exciting these competitions can be. There's still time to participate in the obesity and machine learning competition, and then keep an eye out for, as Caroline said, whatever we cook up next. But with that, just definitely a huge thanks to everybody who put in all this work to make these challenges go and participate today. So thank you.
Thank you.